Hello and welcome back to Microbiology. Today we'll be talking about Chapter 5, which is going to be focused around eukaryotic cells and microorganisms. We are going to start off with a little bit of history about eukaryotes. We've already kind of done this, but I do want to remind you that most likely eukaryotes came a little later after things like bacteria and other single-celled organisms, but around 3 billion years ago would be a good time frame. I do want to point out that these cells, again, came from primitive structures, which include possibly even our organelles originating from primitive cells. So here you can see some basically ancient eukaryotic protists that you can kind of make out features of. The idea is, again, we think, microscopic organisms in the eukaryotic world were the first eukaryotes. But there is an interesting theory that has gained some significant support, and that is the endosymbiotic theory. The endosymbiotic theory suggests that a large precursor cell combined with a smaller prokaryotic cell to create organelles like the mitochondria and chloroplast. This was actually proposed by Dr. Lynn Margolis. So not only did she provide this kind of basically hypothesis that led to a theory, but she provided the support to really help try to validate it. Some of the things that do support this principle, that mitochondria and chloroplasts were most likely prokaryotic cells initially, was again the structures of their ribosomes. They actually have 70S ribosomes, just like bacteria would. Another important thing to note is they have circular DNA, just like many prokaryotes do. They also reproduce independent of the cell and carry out and make their own proteins. And so the idea is the mitochondria and chloroplasts do have all of the hallmarks of what would be a prokaryotic cell that just happens to be living inside of our eukaryotic cell, working together with our cell in a symbiotic relationship. So the idea that, again, initially single cells were the first things that popped up did lead to this idea that potentially maybe several cells grouped together to carry out a very particular function. The idea is, again, several cells carrying out a similar function we call a tissue. And those tissues eventually organized to make organs, and then the organs came together to make organ systems, and then the organism. So that whole idea that, again, life is structured in the kind of... So, whenever we talk about eukaryotes, again, we're concerned with the typically single-celled or microscopic eukaryotic organisms, but I want to stress that we do look at multicellular things as well. So whenever we look at this, do recognize that protozoa are always going to be single-celled. Helminths will always be multicellular, the exception being early in development as a single cell. But then we have fungi and algae that are going to be either single or multicellular, depending on the certain situation or even the organism. So, what we're going to do next is start going through some of the parts that are associated with eukaryotic cells. I want to point out some of these parts are found in all eukaryotes, some are just found in specific ones. So let's start off with the parts found in all eukaryotic cells, like the cytoplasmic or cell membrane. We also see a nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, vacuoles, cytoskeleton, and glycocalyx. Some things that are optional in eukaryotic cells include cell walls, like plants have cell walls, and even some protozoa. Locomotive appendages are also optional, and some cells have things like flagella or even cilia. Chloroplast is the last one that is optional, and something we won't spend a lot of time on because chloroplasts are so associated with photosynthetic organisms, and photosynthetic organisms really don't cause infection. So this is, again, a generalized eukaryotic cell. This is no cell in particular. This is much like the bacterial example where we're going to show you all these features in a cell that really doesn't exist. So if you look at this, again, you can look through the different parts. We're going to spend some time on this. The same way we did the bacteria, we'll kind of work our way from the outer kind of appendages and work towards the middle of the cell over time. And so we'll discuss each of these parts in a bit. So the flagella is really one of the outer appendages that we'll start with here. You've already talked about flagella in terms of bacteria, But there are some pretty big differences between those. First off, the eukaryotic cell is significantly bigger, and it's actually far more complex and is part of the actual cell because the membrane wraps around it. And so the idea here is the flagella is pretty different. Another important thing to remember is, again, eukaryotic flagella can be whipped back and forth or wiggled like a dog tail, while bacterial flagella are rotated, kind of like a propeller. Cilia are going to be another external appendage used for movement. Cilia, much like flagella, help propel the cell. But unlike the flagella, we typically need lots of cilia to end up moving things. So whenever we see cilia, there's usually several of them. If they cover the whole cell, it's likely they're used to move the cell. If they just cover surfaces of the cell, then they're likely going to help push stuff around the cell. And so the idea is these are going to be very similar to kind of like the oars of a crew team, or the rowing action of a crew team, and that we have to work together to create that propulsion or that movement. And so really, just like the crew team example, we see a power stroke which pushes forward, and then a recovery stroke to allow us to do a new power stroke. The glycocalyx is the next topic we should really talk about. The glycocalyx is typically going to be, again, a component found in many cells that is really basically this overaccumulation of usually a lot of sugars on the outside of the cell. Another name for this in eukaryotic organisms is the extracellular matrix, which again happens a lot. We see extracellular matrix even in our tissues. And so the idea is the glycocalyx is something that exists really across prokaryotes and eukaryotes. One thing I want to point out that is different is the concentration of those sugary compounds and the fibers. Again, we see a little more fibers in a lot of tissues in the human body. The cell wall is actually an interesting topic whenever we're discussing eukaryotes. Because it's optional, we don't see it in all eukaryotes. In fact, it's typically fungi, algae, and some protozoa. This cell wall provides the same basic kind of purpose in that it provides support and protection, but overall chemically it's very different. Even in the world of eukarya, each cell wall is going to be unique in its composition. Like plants will use cellulose to make their cell wall, while fungi often use things like chitin or cellulose. So the idea is we get variability in the cell wall structures, unlike in bacteria, where bacteria just really use peptidoglycan. The cytoplasmic membrane is again something we've already discussed. Again, I call it often the cell membrane. The idea is it's a phospholipid bilayer with proteins and sterols embedded in it that helps really control what moves in and out. And as a part of what we'll do later is that discussion of that movement. So it's important to understand that all cells have cytoplasmic or cell membranes, and they all rely on them to really control movement in and out of the cell. The nucleus is again a unique structure to our eukaryotic organisms. Most of the time they do appear to be a compact sphere, and they're pretty prominent inside the cell. It's pretty obvious where the nucleus is in most eukaryotic cells. Really, the nucleus's job is to house the DNA, but we'll often see a lot of DNA and RNA kind of compacted in a smaller region we call the nucleolus. And this is usually where we make ribosomal RNA synthesis. So we couldn't be doing a discussion about eukaryotes without talking about cell division in eukaryotes. And really there are two major types of cell division in eukaryotes: mitosis and meiosis. Really, it's important to remember that most eukaryotic organisms have multiple chromosomes and are often linear. So really, mitosis and meiosis is all about trying to figure out what's happening with the genetic material during cell division. In mitosis, we're going to end up producing an exact copy of the cell, really by going through four major steps: prophase, metaphase, anaphase, and telophase. There will be something that happens in between called interphase, but really, again, the steps of mitotic cell division are prophase, metaphase, anaphase, and telophase. In meiosis, we're not going to produce an exact copy of the cell. Instead, we're going to produce something that is unique in having half the genetic material. So really, we're not going to go through the same steps as mitosis. We're going to do this in a modified version, where we're going to end up doing the basic steps of mitosis twice over. So really we have prophase 1, metaphase 1, anaphase 1, and telophase 1, where we then just go right into prophase 2, metaphase 2, anaphase 2, and then telophase 2. And what should happen then is we end up with half the genetic material, since we went through mitosis twice without copying the DNA again. The endoplasmic reticulum is an important structure that helps provide basically a series of tunnels used in transporting and storage of really things that help make proteins. And so the idea is there are really two different endoplasmic reticulums, the rough and the smooth. The rough is called rough because it often has ribosomes attached to its surface, which leads to, again, the production of proteins. We also, again, store these unfinished proteins so that they can be transported to another organelle called the Golgi here in a minute. The smooth endoplasmic reticulum does not typically have ribosomes, and as such is responsible for producing non-protein things like lipids. The idea here is, again, though, it's a series of tunnels that we can store and transport these products through. So you can see here an image that's illustrating the nucleus and the rough ER. Again, the central point being the nucleus, and then the rough ER. Again, it's rough because of the ribosomes studded all around. And you can see its real job here is to allow for protein synthesis. And so the idea is we copy the genetic material in the nucleus in the form of RNA, and we use that messenger RNA to feed through our ribosomes stuck in the rough ER to ultimately produce polypeptides that end up being proteins later. The Golgi apparatus, we lightly hinted at a minute ago, is the place where, again, we ship those unfinished proteins and products. And really this place is a site for protein modification and really the finishing up of the protein folding. So it's important to understand there are two different sides to the Golgi, basically where the stuff is coming in, where the transitional vesicles come in, and then where the condensing vesicles go out. So what comes in is unfinished proteins from the ER, and what goes out is finished products that need to be now distributed where they go. And so the idea here is we're ultimately using this as a place for protein maturation, modification, even storage, but also a place where we can start to ship things where they belong. So you can see that very illustration here, and that we're making these pre-protein products and we ship them in transitional vesicles over to the basically underside of the Golgi. We finish the protein folding and modification as we move through the layers, and then we package things up and send them to their appropriate destination in these condensing vesicles here. So what we're really getting at is there's overall an assembly line that's formed here. From the nucleus through the Golgi, we're ultimately trying to produce products like proteins. So let's look at that. In the nucleus, we can make things like ribosomes that help occupy the rough ER. We also make messenger RNA that can be fed through the ribosomes to make pre-protein products we call polypeptides. Those polypeptides can be packaged up in transitional vesicles and sent to the Golgi apparatus, where they can then again mature or be modified, but ultimately try to be finished products by the time we leave through the condensing vesicles. Often we see these condensing vesicles moving to the outside and become secretory vesicles, where we can dump again those resources outside through a process called exocytosis. One thing that's not pictured here, but very, very, very important to understand, is notice that everything is moving through in basically a phospholipid vesicle, which means we're naturally moving phospholipids from the center of the cell to the outside. This is not a process that can be continually done without ramifications. Again, eventually we would run out of membrane on the center part, and thus we couldn't continue pushing anything out. So something not pictured but equally important is endocytosis, where we bring resources in to roughly the central point to try to use them during metabolism. So cellular vesicles do exist again in eukaryotic things. Two of them I want to discuss are the lysosome and vacuoles. Lysosomes are actually going to come from the Golgi and contain a bunch of enzymes. And these enzymes are intended to digest things like food and destroy microorganisms and even break down organelles that aren't carrying out their appropriate function. Vacuoles, on the other hand, are typically about storage. We're going to hold things over a long period of time so that we can use them potentially later. So, let's talk about mitochondria. As we've already talked about, mitochondria do share features with prokaryotic organisms. And so really, again, we do think at one time they were prokaryotic. Because again, they have their own DNA, they have their own ribosomes, make their own enzymes and proteins, even again assist in producing product like metabolic products. And so the idea here is these things divide independently, and thus they're probably going to be, again, a prokaryotic organism that are part of our cells in an endosymbiotic relationship. So let's point out, again, we have two major structures here, the cristae, which are folds, and then the matrix, which is the inner part. Whenever we look at this, we can see, again, the very unique structure where we have a bunch of, again, inner regions, but really the folds are what's really relevant here. Those cristae or folds are important in causing or allowing for enough surface area to produce a lot of ATP during something called the electron transport chain. As we said before, the support for the endosymbiotic theory really comes from the fact that the ribosomes are 70S. We have a single circular strand of DNA, and again, the cells divide independently of the rest of the cell. And I say cells, I meant mitochondria. The mitochondria divide independent of the cell itself. Chloroplasts follow the same rules in terms of they have their own DNA, they have their own ribosomes that are 70S, they make their own proteins and enzymes, and divide independent of the cell itself. So again, we also think that most likely this was a single-celled photosynthetic organism that lived in the past. Okay. One thing I do want to stress is at no point do we think that if we pull chloroplasts or mitochondria from the current cell that they will survive, nor will the eukaryotic cell they came from survive. Okay. They have become reliant on each other, and at this point absolutely necessary. With chloroplasts, it's important to understand our primary goal with this organelle is to actually produce, ultimately, enough energy through photosynthesis that we can support the cell. Ribosomes are very important because they synthesize protein for us. They're distributed throughout the cells and are found in all cells because they're necessary. It's important to understand some are free-floating and some in the eukaryote are actually attached to the rough ER. Again, there are ribosomes in the mitochondria and in chloroplasts that are, again, different from most ribosomes in the eukaryote because they are, again, 70S. In the rest of the actual eukaryote, though, we find 80S ribosomes, which are made from 60S subunit and a smaller 40S subunit. And so the idea, again, since there's Fedberg units, they don't add up to what would be their total. So again, 60 plus 40 isn't 100 here. In this case, it's 80S. So I want to point out polyribosomes for a second, because really what ribosomes do is read messenger RNA and translate it into a polypeptide. So if we had a bunch of ribosomes close to one another, we can actually feed the same messenger through all of them to produce lots of polypeptides at the same time. So we can go from little amount of polypeptides and proteins to huge amounts pretty quickly. So you can see the cytoskeleton is actually going to be a very important topic here. There are three major types of fibers that we see: actin filaments, intermediate filaments, and microtubules. Really, their jobs are pretty simple. They help anchor organelles, they help move things like vesicles and genetic material, they help even control shape and movement. The reality is we need cytoskeleton to live, and often it's under observed in terms of graphical images, because if we tried to draw them accurately, we would never be able to see the cell itself because there's so much cytoskeleton. So one criminal thing I think happens is the idea that really vesicles or genetic material float through the cell. That does not happen. Again, they're carried along cytoskeleton, which is a very important concept. So you can see these comparisons of the characteristics of bacterial and archaeal cells or prokaryotes, eukaryotes, and viruses. Again, take and pause this for a bit and look over this if you want to. But overall, most of the stuff we'll cover as we go throughout the course of the semester. So, What we're going to do now that we've covered the organelles associated with eukaryotes is to talk about some features unique to the different categories of eukaryotes we discuss. So we're going to start off by talking about characteristics of fungi. Fungi really come in a variety of shapes, sizes, there's lots of different genuses and species. And so the idea here is we're going to break it up into two groups, but I don't like their macroscopic and microscopic thing here. I don't care a heck of a lot about mushrooms. What I care about are microscopic things like yeast and molds. Okay, and so really whenever I say fungi have two different basic forms here, they're multicellular molds or single-celled yeast. That's what we'll talk about. So the yeast cell is always going to be single-celled. So yeast, like bacteria, are single-celled. But again, these are eukaryotic, so they still have organelles, they still have all the structures associated with eukaryotes, but again, each cell is its own unique individual. One of the important things to understand is that yeast actually reproduce through something called budding. And budding is basically growing a baby yeast off the side. This would be an example of asexual reproduction. I do want to point out that on rare occasion, the yeasts that are budding fail to actually separate. That result in something called a pseudohyphae, which we'll talk about in just a second. Before we discuss again pseudohyphaes, let's talk about a normal hyphae. A hyphae is going to be a bunch of long thread-like cells grouped together to make mold. So again, hyphae are found in molds, not typically yeast. But I just described a little bit ago this idea that pseudohyphae would be a chain of yeast that are stuck together. Again, failures in cell division and yeast can sometimes result in a chaining of yeast, but this is not a hyphae. This is not intentional. That's why it's called pseudo, or false or fake. There are some fungi that are dimorphic, meaning that they can come in either form, as single-celled yeast or multicellular molds. But in general, the idea is many of these things typically fit in one of the two areas. One thing I want to draw your attention to, just for lab purposes really, is this idea that molds can come in a couple different forms in terms of their filaments or hyphae. Notice again there are separations between each cell in a septate hyphae, while in a non-septate all the cells kind of merge together as a single filament. So this was that idea we were discussing about basically yeast budding. So you can see this is the baby yeast that's growing off of the big mother yeast. And the idea is traditionally once the baby or bud gets big enough, it separates and becomes its own yeast. But if we fail to separate, each cell can then start producing its own new bud. And before long, you have a chain of cells. Fungi will actually have nutritional requirements that put them in three different categories. Heterotrophic organisms are actually going to require resources from a variety of sources. And typically, again, they'll take in things like their carbon and energy needs through breaking down things in the environment. Saprobes specifically are actually going to break down dead organic material, so they'll actually gain their food from the dead. Parasites, on the other hand, gain their food from the living, which means, again, stealing it from hosts like parasites do. So here are some examples, again, breaking down things like fruit or organic matter, or even, again, breaking down tissues are an opportunity for fungi to gain resources. Whenever we talk about the microscopic fungi, again, we have the yeast and molds, but multicellular molds really have features that help us in organizing them, including, again, their structures that form things like hyphae, either septate or non-septate, and the ability to form larger structures like mycelium, which is a bunch of hyphae kind of intertwining, making a mass of hyphae. And lastly, their spores that they produce. What features do they use for reproduction? So fungi are ultimately going to have pretty complex and successful reproductive strategies, which have allowed them to become very abundant in the environment. Some of them, again, just keep growing with hyphae. Some of them fragment or break off to make new whole colonies. Some of them, again, do sporulation, where they produce spores that can be spread to other areas. I do want to point out that, again, these spores should not be confused with bacterial endospores. Fungal spores are basically going to be reproductive, while the bacterial endospore is not about reproduction. It is about survivability. So here's some examples of spores, asexual spores. Really the two categories that we care about here are your sporangiospores and kinetospores. Sporangiospores are easy to recognize because they are inside of a sac called the sporangium, okay? While kinetospores will just be loosely attached and can easily come off. Sexual spore formation is actually really important in fungi. Again, sex in general is about bringing two separate parties together to make an offspring that's a hybrid of the two. The idea there is it creates genetic diversity, and that genetic diversity increases the survival of the species. So overall, sex will always be preferred. I do want to point out that we need to identify, often in a medical setting, specific fungi, so it's important to be able to isolate them, look for the presence of spores, including sexual or asexual, or even again try to classify based off of how their hyphae appear, what do their colonies look like, pigmentation, other features as well. So notice again we are giving you two examples of asexual fungal spore options. So you can see on the left here, we can clearly see a sporangium that's holding all the spores inside. So this is clearly sporangiospores. On the right side, notice how there's no sac around these spores. These spores are loose and as such can be knocked off. These are kinetospores. So there are several different diseases that come about from fungal infections. Overall, fungi aren't a great threat to us unless we're basically immunocompromised. That tends to be whenever it becomes a real issue. But they do pose kind of annoyances and things like allergies. One thing that can be a real threat, though, is neurological conditions that are brought on due to ingestion of toxins that come from fungi. And so the idea here is you shouldn't eat any mushrooms you do not know where they came from. The idea being that you could ingest toxins that could honestly kill you. So really, other impacts of fungi include the negative impacts on industry. Overall, huge amounts of food are lost every single year due to decomposition, primarily done through things like fungi and bacteria. But also we use fungi a lot in really improving our environment in terms of breaking down organic material to, again, allow for nutrients to be introduced to the soil for agriculture. Again, taking and basically helping control the association between plants and water and nutrition. Again, production of antibiotics, alcohols, organic acids, vitamins, even beer, wine, cheese. These are all things that come typically through fungal production. So the next category we're going to talk about today is the protists. The protists are again going to be, so algae is something we're not going to spend much time on because again they're photosynthetic, and as photosynthetic organisms they're not typically going to be a huge threat in causing infection. But it is important to know that they come in a variety of forms. They do photosynthesis and produce potentially huge amounts of oxygen, which is again relevant for our environment. Really, plankton are actually very important to the environment, and that they create essentially the primary producer role in the aquatic system. So they're like the grass of the ocean, and so the idea is really life feeds on plankton in the aquatic environment, and as a result, again, very crucial for the food web. They also do produce huge amounts of oxygen for this planet, but I do want to point out something, that they can cause an issue where they produce toxins that can cause health complications. So on to something we do care a little more about in protozoa. Overall, protozoa has roughly 65,000 species. Again, they don't fit super well in a box. We're currently significantly trying to overhaul how we classify protozoa. And it's because some of them are harmless members in water and soil and don't really do anything but help the environment. But there are a few out there that are full-on parasites and cause significant infections in people every single year. And so again, trying to classify these things is a little more difficult. Protozoas do have some structures that are important to eukaryotes, like the organelles. I do want to point out, though, they also have additional features, some of which are similar to things that we know of in multicellular organisms as mouths or digestive systems, or reproductive tracts, or even means of locomotion. So overall, these things will typically come in the two forms: free-living species and parasitic species. Free-living typically scavenge from the environment to eat, while parasitic species again need a host to feed off of. Again, parasitic species are the ones that typically cause our issues in terms of infection. Again, protozoa have different types of movement, and we often, in an informal way, use this for classification. Amoeba move through pseudopods, or false feet. Flagellates move via flagella and can have one or several. And then ciliates move via cilia, which often typically cover the entire surface, creating for propulsion of the cell. Here you can see visual examples of the options in terms of movement. Take a look at them. Again, if you have questions, most of them are self-explanatory. If you see a flagella, you're a flagellate. If you see a pseudopod, you're an amoeba. If you see a cilia, it's a ciliate. So, really, I don't care a heck of a lot about these stages for most of our protozoa, but it is in general helpful to know the difference between trophozoites and cysts, because trophozoites are going to be active and typically in that case cause infection. Cysts, on the other hand, are kind of like an endospore. They're going to be dormant. They're just resting until things become favorable again. And so again, trophozoite is kind of like a vegetative bacteria, and then the cyst is kind of like an endospore found in bacteria. So you can look through the different life stages here. Again, don't really care much more than you understand trophozoites are active in causing infection. Cysts are dormant and often a means of transmission. So on this slide, they're trying to illustrate that means of movement also come with other features. So many flagellates typically have a single nucleus. They form cysts and are free-living. I don't care much about that stuff. Again, I care that you understand that we can have flagellates that move via flagella and ciliates that have cilia. I don't care as much about, one version uses asexual reproduction of fission. So again, that stuff continues here with the ciliates. But I do want to talk about something with the sporozoa. Sporozoa are a non-motile group. Okay, so we haven't talked about them yet because we are classifying based on motility. Since sporozoa are non-motile, they are going to be something that we should probably talk about. And the reason being is because they're all parasites. They don't move because they rely on a host to move them. And so the idea is sporozoa are relevant to us as a group of protozoa because they are parasitic individuals. So overall, again, we do have a formal classification because of features like appearance, but that is something that is going to change. And I don't care a heck of a lot of about this stuff for us and our purposes, but this information's here if you're interested and want to look further into it. So one of the things I want to point out is on this slide, the idea that parasites are often protozoa and helminths. So if you do go into the field of parasitology, you'll often be working a lot with protozoa and helminths. So there are some diseases listed here. Again, we're going to do the diseases stuff later, and so the idea is don't stress about this for the test right now. It would be helpful in the future if you knew about Trypanosoma brucei causing African sleeping sickness or Krusei causing Chagas, but again, right now, I'm not going to hold you accountable for that information. So let's move on to worms. We again call them helminths. Again, these are parasitic worms that we're concerned with. We don't care a dang about earthworms in this class. We're concerned with the worms that cause infection through being a parasite. So really, three major categories we'll talk about: tapeworms, flukes, and roundworms. Again, they'll vary in size, they'll vary in location and where they infect. Again, overall, the one consistency in this group is they are parasites. So let's point out again, worms are multicellular and they are animals. So they're the most like you and I in terms of the things we'll discuss out there. So they have reproductive tracts, they have digestive systems, nervous systems, muscular systems. They are animals, just like us. So one interesting fact about them is they do have complete life cycles where they start off as a fertilized egg, go through a developmental stage, and then an adult stage where reproduction is the purpose. And so the idea is they have similar life stages to even what we have as humans. So nematodes, trematodes, cestodes, again, they're helpful in the classification of really worms, but for us, we're not going to do much with them. I care a little more that you look at the word hermaphrodite and understand that that means that an organism has both male and female sexual organs. So I want to point out towards the bottom here the general life cycle though. Transmission is usually of an egg or a very young worm, where it then grows into adulthood inside of its new host. And for us, we often are that ending point, the final host, where again the worms will start to lay eggs to try to move on. So overall, most of our exposures come from primarily food, soil, water, and really animals, which again can go back to that food idea. And most of the time, worms are taken in through oral ingestion. There are some that come through penetration of unbroken skin, but they're very rare, and we don't see a lot of them here. Again, those worms in particular have very well-defined mouth parts to allow them to eat through your skin. Ultimately, again, we are going to be a place where these things settle, but overall in nature we find worms seen in animals or insect vectors that can carry them. So let's actually take some time to talk to you about an example of a worm here with Enterobus. Enterobus is more commonly known as the pinworm and actually causes really just infection of the large intestines. Overall, they're pretty small, hence the name pinworm, and usually don't spread and cause serious infection at all. But what's interesting about them is their technique in actually reproducing. So again, we have the examples of a female and a male worm. Again, we can have essentially the production of a fertile egg. And what's interesting about this egg is often released on the anus at night. And so what happens is that egg causes anal irritation, and then ultimately scratching happens, causing now a spreading of the worm to the hands. So now whatever the individual touches can now have the fertile egg on it. So all it takes in is an action that moves the worm egg to a person's mouth before they're infected. And so the idea is this thing is really found primarily in children, whom again itch their anus in response to the eggs being laid, but then can allow for self-reinfection by putting their hands in their mouths or by touching things that other people then put in their mouth. And so again, this is a childhood style disease. It is slightly gross, but overall it's not dangerous. Again, if a child does have this, it's very important that we try to clear that up just to make sure, again, they're as healthy as they possibly could be. So overall, there are formal and informal ways to classify these worms. Again, shape, size, degree of development, what organs, infects, that kind of stuff. But again, I'm not going to be a huge stickler on the classification of worms. Again, identification of these things are typically pretty easy because they're worms. You can usually visually see them without much aid. But if we do need some, microscopes are available to help us with that identification. So this slide's really trying to illustrate to you the fact that there aren't huge numbers of different worms that are parasites to humans, but typically there are huge amounts of people that have these infections. And overall, we do expect this to be a very, very, very common problem. Again, yearly estimates of worldwide cases are expected to be in the billions. Even somewhere like the US, where we expect most people to not have worms, we still expect around 50 million infections each year. So the idea here is worm infections are pretty common. And through history, again, they were super common. Really, 100 years ago, you couldn't really be confident that you didn't have worms. So what I'm getting at is this kind of idea that our existence without having worms in us is a relatively short time, that really we've evolved with worms. One thing I want to kind of introduce here is this idea that since we've not had these worms inside of us, we've seen a huge increase in autoimmune disease and allergies. And so there's a hypothesis out there that has been gaining ground that illustrates that there's a chance that failure to have worms as part of our biota can actually result in things like autoimmune disease or allergies. This is still very, very early, and again, should not be taken as absolute truth. But there may be a day where you say you have Crohn's disease, issuing a worm for you to take as a supplement may actually treat and fix issues associated with Crohn's. And so what we're getting at is autoimmune diseases today we don't have a lot of answers for, but one of the things that we think may play a role is our basically trying to eliminate the things that are normally part of us. So this table here is really trying to illustrate the differences between things like protozoa and fungi and algae, helminths and humans. Again, this is trying to give you some quick visual references to differences, but again, this won't necessarily be a topic that I stress in terms of being a major testable topic.